Okay. So we're here with Brian Culley, the CEO of Lineage Cell Therapeutics. And I'm really glad that my followers reached out to me and told me to research Lineage Cell Therapeutics because your OpRegen platform is right in my alley. So, there's so many advancements being made in clinical trials, so it's important to stay on top of them. Sometimes we see patients with such extensive retinal damage in a variety of indications, but I have conversations with different ophthalmologists that have been in the field for quite a bit that they think this technology just sounds way too good to be true. And another really cool thing is that it's also interesting is that my listeners includes a wide spectrum of retail investors. So some with a lot of knowledge in medicine and science and some with little to no experience in medicine and science. So I really hope that everybody will get something out of our conversation today. So Brian, obviously this isn't your first rodeo. You've had prior experiences at Artemis and Mass Therapeutics. So can you tell us about anything you learned at these companies prior to your arrival at Lineage?
Oh, wow. I think over 15 years of running public companies, there are so many lessons that you learn. One thing that I guess I could say has been reinforced for me is that my favorite aspect of our industry is running clinical trials. If you think about companies as having 3 stages of their life cycle, from the early research through the clinical development and ultimately commercialization, where I'm happiest and probably do my best work is where we are, which is in the clinical development, because it really helps me bring my scientific background and my business background to bear in one place. So it's been such an amazing ride to be responsible for advancing products through clinical trials, and I'm just excited to keep doing it.
Awesome. And Brian, so before we get into OpRegen, what would you say is the broad mission of Lineage Cell Therapeutics?
Well, ultimately, what I think the duty of Lineage is to help usher in what we see as a new branch of medicine. There were many promises made 2 decades ago and more recently around cell therapy, and they continue to be promises around cell therapy. And there's been a lot of hope and a lot of activity, but it hasn't led to as many successes, commercially available products as all of us would have expected or have liked to have seen. And now maybe it was a little too ambitious, or maybe it wasn't mature enough. But I think today the industry as a whole has learned a lot about where cell therapy is best deployed and how to solve some of the key problems that are in the field. And so Lineage, I believe, is very well positioned in areas that have got higher probabilities of success than some of the prior endeavors in cell therapy. And, I love our particular spot of the world. So I really want everyone in the organization to be aligned with the mission that, we have an opportunity to break out a whole new branch of medicine, to be pioneers, to be historians, and lead this charge. And it's an exciting responsibility.
Awesome. So you mentioned you want to be like pioneers. So I'm curious, what are the benefits of Lineage Cell Therapeutics pursuing pluripotent stem cells in comparison to a program that perhaps focuses something on like multipotent stem cells?
Well, so stem cells come in a bunch of different flavors. We like pluripotent stem cells because from them, as starting material, you can manufacture any other cell type. I mean, every cell type in the human body used to be an earlier stage, undifferentiated cell. So you're able to, I'm going to say, always run the tape forward. So if you want a liver cell or a kidney cell or a bone cell, you start with a pluripotent cell and you perform activities to that cell in order to turn it into or differentiate it into the specific cell type you want. Now, when I say only running the tape forward, there are other technologies that can take mature cells and reverse them, but that requires a considerable amount of genetic manipulation. And we don't really know as an industry what the impact of those manipulations will be. And so I'm not particularly interested in using adult cells and reversing them to make them multipotent or pluripotent. I really think it makes a lot of sense to start with a pluripotent stem cell, and then from that derive the specific cell type that you need, because I think fewer genetic aberrations are likely to result.
Awesome. And so, though I'm just a young optometrist, I've spent many years working for retina specialists during my college days. So unfortunately, I know how common the condition of age-related macular degeneration is and how debilitating AMD can be. So could you explain for the viewers what exactly is AMD, and why the buildup of lipofuscin is such an issue? And in addition to that, why is the ability to restore retinal tissue so unique?
Yeah, so the finding, the discovery, and you use the phrase "too good to be true," and I love that because I find it really flattering, because the evidence that we have is anatomical evidence, right? It's not hearsay. It's not how a person is feeling. We're actually looking at physical pictures in the eye. So to explain that, the underlying condition of dry AMD, the prevailing feature of that is the death of specialized retina cells. So in the back of the eye, as you know very well, but just to kind of collect some folks who might not be as familiar, the back of the eye, the retina plays a vital role in your ability to see things. And the retina is comprised of about 12 or so layers of specialized cells. And there's a layer of cells back there called RPE cells. And those cells, for reasons that we don't fully understand, can begin to die off. And this typically happens with age, although there may be genetic factors, there may be environmental factors. But, if you live a really long time, these cells get tired, they work very hard, and they can begin to die off. And we really don't know the reason why they die off. But when they do die off, you lose your vision. And typically how this occurs is just one little tiny spot, one small area. We'll call it an area of atrophy, geographic atrophy. And that spot will get larger and larger. It's almost like one of these wildfires that just kind of expands outward. And so eventually that spot will get large enough and wide enough that it affects an individual's vision, and most typically their central vision. So this is your ability to see faces or, focus on your cell phone. And eventually this can lead to total blindness because these cells will just keep dying off. And unfortunately, human beings do not possess the ability to regenerate retinal tissue. We can't regrow tissue and heal this wound the way that we can if you cut your arm. So the Lineage approach is different. And it's different because the traditional methods usually tackle just one pathway. That's how the pharmaceutical industry and biotech industry usually behaves. We see one problem and we come up with one solution, and that's science. But in the case of dry AMD, there are probably thousands of problems in these cells, because the cell is dying, right? By the time a cell dies off, there are so many things going wrong that our view is that the only way to fix that is to replace the entire cell. So we manufacture billions upon billions of brand new RPE cells, and then we transplant those cells into the eye to take over the role of the ones that have died off. And in doing so, we have seen really exciting results, including improvements in vision. We've seen these anatomical changes, but the one thing that has been really extraordinary is what we call retinal restoration, which is we are seeing areas of atrophy that are devoid of these RPE, devoid of the most integral tissues. And then after treatment, those areas are restored. And so that is what's really incredible, because as I say, humans are not starfish. We can't regrow an arm. But we have shown that our therapeutic intervention has been able to restore retinal tissue. And there are images. You can just look at the anatomical changes. And the retinal experts that we have had review this data are very excited about it. And so obviously the rest of us are as well.
Right. I know I'm really excited about it. And I'm really excited to go a little bit deeper into the subjective and objective improvements for the patients and what they've witnessed and what they've felt. But just before we get into that, I had a question for you. I wanted to expand on AMD a little bit. So what does the wet AMD market look like compared to the dry AMD market? Because we know we have two types of AMD, correct? I think we only touched on the dry AMD portion. So why is it so clinically relevant to target dry AMD perhaps than, targeting wet AMD? What's the importance of targeting dry AMD before we get to that wet stage?
Yeah, that's an important distinction. Oftentimes people, when they're speaking to us, they sometimes get confused about wet AMD and dry AMD, or dry eye, which is a totally different thing.
That's funny.
Right? So within AMD as a whole, the two forms that you correctly reference are wet and dry. So the hallmark of wet AMD are these leaky vessels. So we understand very well on a molecular level what is going wrong in the setting of wet AMD. And so we as an industry have been able to develop drugs, and these drugs are wildly successful. The wet AMD market is doing north of $10 billion in sales. So as an industry, we've done an incredibly good job of coming up with successful therapies to treat wet AMD, but we have nothing for dry AMD. There are literally zero approved therapies. So we have incredibly 8 to 9 times more individuals with dry AMD than we have with wet AMD, but we have nothing for them, right? They are told things like, don't smoke, eat leafy green vegetables, be healthy. That's not therapy. So you have this huge unmet need. And our view of the explanation, like how can it be this massive market? Our view is that when you look at the failures are all what I said before. They are single mechanistic targets, trying to hit a few pathways at once, but there's so much going wrong. It's just sort of too late to intervene in that way. And maybe you need to come at the whole problem with an entirely different approach. And then finally we can be successful. And that's really what cell therapy is about. That's what I talked about with a new branch of medicine, new approaches, new methods to make progress where heretofore we have nothing.
Amazing. It was really well said. And it was really important to discuss that distinction, I thought. So your clinical data showed that after 3 years post-OpRegen treatment, you were able to slow the growth of geographical atrophy and the satellite lesions. Visual acuity of the treated eye remained steady compared to the baseline of the untreated eye. We saw some evidence of retinal regeneration on the OCT, whereas again, the fellow eye followed the predicted course of lesion growth or degeneration. Could you go into some detail about what you observed objectively and also anecdotally from your trial participants?
Yeah, I think that's an important continuum. I mean, one of the things, just to go back to your first question, one of the things that I've learned over time in this industry is that it's really easy to find a single assessment or a single measurement that kind of tells you what you want to hear. But if you want to believe in an approach, it's far better to see a whole host of assessments all kind of directionally going the right way, right? So in our case, what are the kinds of things that we want to see? Well, we want to see both anatomical changes that are consistent with what we think the mechanism of the treatment is, and we also want to see functional changes. And this, on the functional side, has a number of different categories. So I'll hit a few of them. One of them, the easiest one to understand by far, is how many letters can you read on an eye chart, right? They have the ETDRS. You've probably administered this 1,000 times. It is a representation of how well you can see, right? Very easy for everyone to understand by reading letters. And we have seen in the majority of the treated eyes following our therapy that people can read more letters on an eye chart. Okay, that's great. And also at the same time, the untreated eye, the one that we don't help, that eye has gotten worse and reads fewer letters. So, all right, that's a nice assessment, and directionally that's encouraging. But then there are also other factors, right? There's things like low light reading speed, or, other quality of life factors that sometimes come through patient reported outcomes, people who worry about falling down stairs or crossing streets and things like that. We've also seen that our treated patients are reporting higher scores on quality of life tools. The other third area, which is really exciting, is something called microperimetry. So now we're starting to kind of bridge between function and anatomy. What microperimetry is basically reporting, being able to see little flashes of light. And one of the things that we have seen is that in an area that was previously atrophic, and now we have restored, that we have seen evidence of greater light sensitivity. So that's really important, because one of the criticisms that you could levy at us is that we're putting material there, but how do we know it's functional? That's a fair question. Well, what I would say is, how do I explain that people are reading more letters? How do I explain that people are happier? How do I explain the microperimetry data where they're registering that they are seeing a flash of light? You can't fake that. You either see that flash or you don't see that flash. Right. And then moving on to the anatomical side, this is where you shift from subjective measures to more objective measures, meaning the patient does not have the ability to influence the outcome. Someone who leans forward in their chair when they're reading the letters, right? They can influence the outcome a little bit, or they could maybe, get used to using a different part of their retina, right? That's one of the problems with those subjective measurements. So for us, we really embrace and rely even more on the anatomical measurements. And so what we do here is we use multiple modality methods. So fundus autofluorescence, or FAF, is one of the very standard and easy to use methods where you can take a picture of an area of atrophy. Another one, which is a little bit more refined and gives you more information, is optical coherence tomography, or OCT. This allows you to essentially see a cross section. And that's where we're seeing this retinal restoration. That's where we're seeing, if you think of this as like a 7-layer cake, right? All these little layers of the retina all stacked up. As you scan through and you move into the area of atrophy, these layers sort of collapse down. And that's how you know you've entered the area of atrophy. Well, we can go back to the same area, and we know that the anatomy is unchanged because you can orient these images based on blood vessels which don't move, and you can see that they've like inflated these areas. In particular, what's called the ELM. All of the most critical pieces of the retina have been restored. They're present. You can literally see them. No human being has the ability to increase or diminish at will the amount of retinal tissue, right? It's a picture of what's going on. It's like, testing white blood cell counts. I can't influence it. It happens. There's a number. It spits out from the machine. So all of this all together, the anatomical, these structural changes, reduction in drusen, all of these to us are arrows pointing in a certain direction that says that our intervention is leading to positive clinical outcomes in these patients, and give us a lot of excitement about continuing to develop this program.
Right. Amazing. That was a really good transition, because I did want to discuss, maybe, do you find that there's a gold standard to prove objective improvement for AMD? Because we know there's fluorescence angiography, we have OCT. So do you find that OCT is one of the best modalities to prove objective improvement? And I know you mentioned the ILM was one of the layers that improved. Did we see specifically improvement of the photoreceptor layer and the RPE? Did we see those layers regenerate as well? Because as we stressed, these layers are not supposed to regenerate. Once these layers are dead, they're dead. They aren't supposed to come back. So, seeing these layers regenerate, it's like we've never seen anything like that.
Yeah, no, I think, that's why we're excited about it. I think Curcio probably did the best work on demonstrating that the ILM, ELM, the photoreceptor and RPE layers, that is what's most intimately linked with activity, i.e. vision. So being able to see those areas of restoration restored is very exciting for us. What has been interesting is wanting to move from FAF to OCT. So I had just anecdotally, what I can tell you is that we had a time not long ago where a couple of competing approaches at some other companies, that they had some KOL days, right, where you bring in some famous doctors and they talk about, the programs. This is routine. Every company does something like this. But it was really remarkable that both of those leading companies in the space had their expert physicians who were talking about how OCT is much more powerful. It's a better way to go because it gives you nearly a histological level of evidence, right? You can see every single layer, whereas the FAF is sort of like just an aerial shot. And so, it's anecdotally, these things matter. We've got these great patient videos where people talking about their experiences. But again, I always come back to the anatomy. The anatomy, to me, these images are irrefutable. And so being able to say, hey, look, here's a before and after. And by the way, I should probably add that these patients that we have reported seeing this retinal restoration in, all of them were confirmed that their areas of atrophy were growing previously. So it's not that somebody, magically just, stopped growing and we just took pictures of them as they no longer changed over time. They were confirmed as having areas of atrophy getting larger, and then we actually made them smaller. And only after 3 years has this one individual gotten back to baseline size. That's unprecedented. It's the right word for it.
Right. So I definitely noticed that you went for more difficult patients. And I thought that was very impressive. I did want to ask, so what were some of the outcomes you observed when comparing slow growing geographical atrophy, like soft drusen, for example, versus faster growing geographical atrophy, in patients with reticular drusen, for example? That was a really good transition.
Well, the clinical study so far has only been in 24 patients, and the first 12 of them all had very advanced disease. They had large areas of atrophy. They were all legally blind. So it really, those first 12 were sort of just the safety component of the study. And then when we moved into the second 12 patients, these are individuals who more accurately reflect what we would envision to be our intended patient population. So they could have vision as good as 20/65 and on average had smaller areas of atrophy. But now you're still only working with 12. So it's really hard to start to sub-analyze the data when you get down into smaller numbers. But what I can tell you, and these are familiar terms for you, but there are these things called IRORA, IRORA, which essentially are tiny spots or flecks of atrophy, which haven't made it all the way through the retina yet. They are immature, like, areas that are starting to sort of, rust out, and what we were able to do is eliminate them. And that's really incredible. I mean, to be able to just kind of, wipe these things out, to eliminate these IRORA, because the IRORA, the I stands for incomplete, they become CRORA, and the C stands for complete. So again, this is a unidirectional condition. It only gets worse. And even some of the competitors that are out there have been really excited that they say, oh, look, we were able to take some of these IRORA and slow them from becoming, go from incomplete to complete. And I agree, that's a good thing. But what's a better thing? Eliminating them completely. So when we think about our patient population and where do we intervene, I think we're going to learn the answers to that as we go forward. But generally speaking, I think that earlier in the disease, when you have a smaller area of atrophy, is probably better. But I probably wouldn't mess with someone who has 20/20 vision. Right? If you have not had the part of your macula that is responsible for vision has gotten involved yet, and you still can see pretty well, even if you've got a pretty good sized GA, not sure you're really the best patient for us. But we may get there. We may get to the point where this procedure becomes so routine and safe that, maybe people with perfect vision will have an intervention to stop the growth of atrophy before they start to lose vision. It's a little aspirational, but I mean, you have to be.
Right. I just want to add for the listener. So, what's indicated for early stages of AMD is something called Arid. So it's sort of like a formula of different vitamins and things, which sometimes helps, sometimes doesn't really help. It's been shown to slow progression just a little bit. So I thought that was really important that you hit on the point of when is it too early to intervene, because sometimes you're putting patients at more risk. So we have to, weigh the pros and cons of treating these patients with Oprogen. But
hey, Nat, I got to tell you, my dad was on the Arid study.
Mm-hmm.
So,
was he?
Yeah,
it's amazing stuff.
Yeah. Small world.
Very cool stuff.
Yeah.
So you mentioned on like the IRORA. Could you explain a little bit about transition zones? Because I know that's a big part of what you're studying. So could you explain transition zones and as targets for the therapy surrounding the geographical atrophy? And is Oprogen capable of restoring the tissue more centrally in location of the geographical lesions?
Yeah, that's a nice question. So I don't think it is. So if you think of the area of atrophy as roughly circular, imagine as sort of like a desert among transition growth, right? So the area of atrophy where we place our cells, where it's most powerful and where we've seen the benefit, is in what's called the penumbra, or just the perimeter, right? That's where the war is being waged. That's where you have sick cells, but they haven't died off completely. So that's where we view that we've got some rescuable material. In the middle of the atrophy, that's a wasteland, and planting some trees there, they just don't have the environment to survive. So I think that in the distant future, if someone has a large area of atrophy and wants to have some kind of therapy, perhaps there will be some sort of, total retinal replacement strategy. But frankly, I hope no one ever gets to that level, right? I want to intervene before anybody gets to that situation. But for us, what we know is happening is that there is going to be earlier and earlier detection of these individuals, because the technology by other companies is in place to be able to find patients sooner. So I think that we're going to be able to identify, you're going to have someone who walks into like a a drugstore and sticks their eyes against a little scanner, and it's going to say, oh, you should talk to your optometrist because you may have AMD. And that person might have perfect vision and would never have known it, right? I might have AMD right now and have no idea about it because my vision is pretty good. So I'm pretty excited that we might be able to get into a situation where there's early intervention, that it's one-time intervention. We stop these little things before they get out of hand. And these transplants, because they've never been rejected. We've never had a patient reject these cells. Hopefully they will last for as long as the individual survives.
Amazing. So Brian, through your clinical studies, have you encountered, have you decided what your best administration modality probably is? So I know I've read about like suprachoroidal versus subretinal injections. So what did the data show to be more effective, and what do you think you're going to be pushing forward with?
Well, with many things, there are trade-offs. So the way that we initially started the program and the way that we're using it now is a straightforward pars plana vitrectomy, which means we remove the vitreous, and then a retinotomy. So we, colloquially, we poke a needle underneath the retina, and then we deposit the cells and we remove the needle. That's pretty good because everyone who comes out of retina surgeon school has to learn how to do that. So it's a pretty straightforward procedure. The whole thing takes about 30 minutes. We have patients that are out of the OR before our staff is out of the OR. So it's a pretty straightforward approach. But we did pilot another approach, a different approach called a suprachoroidal approach, where you go around the back of the eyeball and you come up underneath the retina. So it's just accessing the same the same subretinal space, but you're doing it from below instead of from above. There were some things in terms of the flexibility of that approach that we felt that we couldn't sacrifice, because one of the things that we saw clinically in these patients that had the restoration was they got very complete coverage. And so the flexibility of pars plana vitrectomy plus retinotomy allows us to place the cells kind of anywhere. There was a little bit more stringency with the other device that didn't allow us to get kind of where we wanted every time with our placement. And so we moved away from it. But I will say that we are always going to continue to be open, because we know this is a new technology, and new technologies get better with time. So I think we will continue to evaluate and consider different ways of administering the cells, different ways of handling the cells, and just develop protocols that get better and better, and lead to better outcomes for the individual.
Super cool. So what significant adverse effects have been observed with OpRegen, and do the benefits of OpRegen treatment seem to outweigh the risks? I'm aware of instances of epiretinal membrane, macular hole in one patient, potential for retinal hemorrhages due to the injection, or any other complications with immunosuppressives. Does anything, does anything stand out to you? Anything that maybe worries you, or do you think the trade-offs are pretty good?
Well, you're talking about vision, so people will tolerate an awful lot. But most of the adverse events were associated with the immunosuppression. The immunosuppression used to be 12 months. We've got it down to 90 days. And actually, we had a patient who didn't have it's called tacrolimus. They didn't get any tacrolimus. We had another patient who developed COVID and took a holiday from the immunosuppression. So I don't know how low we can go, but we've moved it from 12 months to 9 months, and we've never had a single case of rejection. And we've got people who are out 5 years now. So I think on the immunosuppression side that, directionally we're going definitely in a good direction. Procedurally, you've got a couple of things, right? You've got a very fragile area, right? You're talking about someone who's maybe, 75, 80, 85 years old. And as soon as you stick a needle into the eye and start poking around, you're bound to have some events. Epiretinal membranes were one of the most common adverse events or observations that we had. Most of those were not clinically significant. We saw them, so what? They're not getting any worse. We did have one that we intervened. We had an epiretinal membrane that was interfering with the patient's vision. So they had a benefit of vision, then they developed a large ERM. We surgically went in and removed it, and then they bounced right back up to the better vision and actually had that sustained for a very long time. So I think there will always be some rate of adverse event that we don't want, and we hopefully will drive that rate lower all the time. But we have demonstrated that you can have an epiretinal membrane removed and still have a very good clinical outcome. Retinal detachment is always a risk. CNV, choroidal neovascularization, or the development of some leaky vessels, that occurs. We had one, but I mean, it was 2 years after the patient was treated. So probably they were going to develop wet AMD without us. So I don't think that that's a high risk for us. There will always be some level of adverse event or risk with any invasive procedure. But again, I think when you talk to the individuals, and we have a couple of patient videos where people talk about their experiences being on our clinical trial. When you talk to these individuals about what it's like losing their vision, I think we're really in an attractive place with respect to the risk reward profile of OpRegen.
Right. And I like to tell my patients, anytime anybody ever tells you that, a treatment has no risks, or, you'll be, there's no risk at all, that's a total lie, because everything comes with a certain amount of risk. But luckily it sounds like the adverse effects that we're mentioning here are things that these doctors see quite often, and performing these procedures to combat the adverse effects are just, typical procedures they see, day in and day out, So Brian, do you foresee breakthrough designation for OpRegen? So I just want to add for the listeners here. So breakthrough designation is the expedited development and review of drugs that demonstrate substantial improvement over available therapies on clinically significant endpoints.
Yeah, certainly. I mean, I think we qualify. There are a whole bunch of regulatory designations, things like fast track status, breakthrough designation, priority review, accelerated approval, regenerative medicine designations. Each of these has its own sort of set of benefits, and they each have sort of their guidelines for when you seek them out and obtain them. So 2 of them that I think are applicable to OpRegen are RMAT designation, that's a regenerative medicine designation, and breakthrough designation. So those are 2 the boxes that we haven't checked yet, but I expect that we would look to get both of those designations. So if anyone's wondering whether we had sought those and been rejected, that's not the case. They just haven't hit the to-do list yet.
Okay. And now I want to pivot to another Lineage Cell Therapeutics indication. So that's OPC1 for spinal cord injury. So could you tell us a little bit about this program and the data points you've observed, such as objective improvements on MRI, motor function, things like that?
Yeah, this is a really powerful one. So conceptually, it's the same thing. We manufacture a special cell type and put it into the patient. But in this case, the patient has suffered a spinal cord injury. So this is, typically a young person, car accident, diving board, motorcycle, whatever. And there's an area of damage and the cells die off. So just like in dry AMD, where the RPE cells are gone, the spinal cord cells are gone. So we manufacture cells called oligodendrocyte progenitors, and they're responsible for manufacturing the myelin, or the insulation for axons. So you may have heard about people going after spinal cord injuries to try to improve mobility, and trying to grow axons. It's not that hard. There's a lot. I used to do this when I was in a PhD program. There are a lot of different things that you can do to make axons extend neurites. But if they're not insulated properly, if they don't wire together, they won't fire together. So you don't get sort of coordinated motion, and you get like spastic movement and pain and all sorts of things. So what we do is we manufacture oligodendrocyte progenitor cells, and we transplant them right into the spinal cord. And the reason for that is a hole would otherwise develop, a cavity would occur. And so we fill that cavity with the same type of cells, which manufacture the same material that your body would manufacture. And we sort of spackle that space and we fill it in, because the electrical impulse that travels from your brain to your hand, it can't jump a gap, right? Electrical current can't jump a gap. You have to have something in there. So we put the material that belongs there. And what we have seen when we've treated 25 individuals with spinal cord injuries, a third of those individuals gained 2 or more motor levels on the gold standard score. So you're talking about, for example, we had a kid who all he could do was shrug his shoulders a little bit. Everything else, he was paralyzed. Now he can type 35 words a minute. Being able to move your wheelchair right? Just that little bit of mobility is life changing. And so we're focused on these injuries, the cervical, 4, cervical 5, 6, 7 injuries, because those are the ones that weave out to your upper extremities. So we're trying to get people to have upper extremity mobility gains after their accidents so that they can have much higher quality of life and not rely on someone else to, feed them, bathe them, and help them go to the bathroom.
Amazing, super cool stuff. What opportunities do you foresee for, perhaps maybe, because you mentioned, Brian, acquired spinal cord injury. So do you foresee any other opportunities for like demyelinating conditions? Perhaps maybe something that didn't happen through like an accident?
Yeah, that's a subtle but kind of cool point. So, people tend to think of the company, they're like, oh, they're working on dry AMD. No, we manufacture RPE cells. What else could you use RPE cells for? Could you use them for Stargardt's disease? I would expect so. And it's the same thing with the OPC1 spinal cord program. What we do is we manufacture cells that produce myelin. So you could conceive of us using it for other conditions for which the feature is demyelination. And so you've got things like, optic neuritis, multiple sclerosis, white matter stroke. I mean, it's like there's this long list of things. Now, resources. We got to focus and, make sure that we're doing one thing at a time. But I believe, and this is my view of how the company can grow, as we stepwise reach these important milestones and we have success with our initial areas, that will enable us to acquire the resources to broaden and to go into other areas, sometimes using the same cells and the same material, sometimes going into whole new areas. So there's a long-term sort of strategy, and people joke about this, I guess, but I say it's like being Amazon. Amazon used to sell books and now they sell everything. Well, you've got 200 different cell types in your body. Right now, we're doing retina cells, spinal cord cells, and dendritic cells. I got 197 ideas that I need to work on and think about. So that's kind of our story.
Right. And could you explain the importance of receiving orphan drug designation for this indication and also receiving RMAT? So RMAT is regenerative medicine advanced therapy.
Yeah, RMAT gives you, more access to FDA. You don't just pick up the phone and chat with FDA anytime you want. There's an organized process and there's calendaring and everything. RMAT designation gives you more access to and more meetings with FDA. So that's an important designation. Yeah, and we have that. Orphan designation is a little different. Orphan designation, the biggest advantage is it gives you some privileges with respect to exclusivity, market exclusivity. So if you were manufacturing something that you didn't have patent protection, you could still get 7 years of protection by having orphan designation. Now for us, we've got some very fresh patents. I'm not really worried about market exclusivity, but it is nice because having orphan designation does give you some other regulatory advantages. And again, anytime you are able to highlight, as we do with spinal cord, that there are no alternative therapies, right? There is no treatment for these patients. It does give you a little bit more, influence to be able to kind of, demand attention and focus down and let's help patients that have no options. Right.
I thought it was really funny when they called them your too good to be true indication because it's just amazing stuff. Brian, how important is the delivery aspect for indications like spinal cord injury? And what are some of the proprietary delivery systems or surgical equipment Lineage is using?
Yeah, I think delivery is critical. I don't happen to believe that you can put stem cells in a patient and they're going to work at a giant distance. What I do believe is that if you use differentiated cells, in our case, spinal cord cells, more specifically oligodendrocyte progenitor cells, that you need to place them right in the exact spot, that you can't put them at a distance. So placement is critical. It's an important component of our therapy, and it will be directly linked to outcomes. Now, in the original study, there was this giant scaffold and this equipment that was used in order to deliver the cells, but it was clumsy and difficult. What we're demonstrating, right, and we're going to demonstrate in a small study, and I hope that it starts around the end of the year, is we have this little contraption now that just sits right on the patient. It still has an XYZ manipulator. It still is just a needle that pokes down in the spinal cord. But the magical thing here is that it allows you to deliver the cells while the patient's connected to the respirator. So the patient's chest cavity is going up and down. but the needle's going up and down the same way. That isn't how it used to be, right? It used to be unplug, hurry up, get the cells in, right? That's terrible. You don't want, does anybody want to rush a surgeon, right? Like, don't do that. So having this device where maybe we'll take 5 minutes to deliver the cells instead of 1 minute, I think that's going to introduce some safety. And we have other advantages in this program. We don't have dose preparation. We reformulated the therapy so that you don't have to wash the cells beforehand. You just thaw them out and they go in the patient. It takes 5 minutes. Okay. So these are important commercial components, which is part of our mission to mature the field, right? It used to be really clumsy. I can get by and do a dozen patients. But if you want to do, 12,000 patients, you got to make it more user friendly.
Right. So I just want to confirm, can you dose the patient at a constant rate? Is that what you're implying? That the cells are delivered at a constant rate?
Yeah, I don't think it has to be at a constant rate, but here's the thing. If you want to eliminate the dose preparation, right, which, that's a source of error, that's a source of contamination, you would literally start the day before plating out the cells and washing them and harvesting them, all that. I want to eliminate all of that. So I would just want to have them in a frozen vial. In order to do that, we had to increase the volume. But the volume of the spinal cord, there's only so much material you can put in a spinal cord, okay? So we need to put it in slowly. So there we ran into the problem where if you unplug someone from a respirator, you can't go slowly. You have to go quickly. So we solved that by coming up with this new device that's attached to the patient. Now we can administer the cells in our larger volume, more slowly, more carefully and safely. And again, we skip 90% of the cost and confusion associated with all the dose preparation. We literally are taking the cells from a frozen state, thawing them at the bedside, getting them into the needle and putting them into the patient. So a much better way to go. So we're going to try and validate that device in a small clinical trial while we're preparing for a larger efficacy trial, which I expect would begin next year.
I love that. So sometimes people forget that we have the therapeutic and then we have the delivery modality, and they have to work in harmony and they both need each other. So it's really important to have both and both working really well. Mm-hmm. So last but not least, Brian, could you briefly explain the VaxBack Platform and the potential for multiple product candidates?
Yeah, this is really a platform within our platform. And that's because we manufacture a third kind of cell. We manufacture a dendritic cell. And this is part of your immune system. And immunology is complicated and I won't dive into it. But what dendritic cells do is they carry a message to your immune system. So this is a vaccine. You can think of it that way. The dendritic cell itself is just a carrier. It's just a messenger. We can put any antigen, that's the message. We can put any antigen onto the dendritic cell. And the antigen is what defines the product. So for example, let's say you wanted to have a product against an ovarian cancer and you had an antigen that identified ovarian cancer cells. You could put that antigen on dendritic cells and put 50 or 60 million of those cells in the patient, and their immune system is going to respond to that antigen. And then if you said, well, okay, I'm done with ovarian cancer. Now I'm going to go to, chronic lymphocytic leukemia. You might have a different antigen and you put that different antigen onto the dendritic cell. So we, as the manufacturer of the carrier, have the luxury of being able to develop our own antigen dendritic cell combination. And we have that right now in a form of lung cancer. But we could also partner with others who have antigens, and we can use those antigens, whether those are famous academic sites or other companies, and we can develop new products where someone else pays for the development and we just take a bite at that apple. And we actually announced that recently. We did our first deal of that sort with a company called ITI that has a glioblastoma or brain cancer approach. They have an antigen. They're going to piggyback on our delivery system, and we're going to develop a new therapeutic for brain cancer. And it's a great way of expanding. And in theory, you could have 100 different cancer programs just from using dendritic cells to carry those messages.
Amazing. So Brian, I'm really grateful that you joined us tonight. I think we had a lot of informative information. The chat was really great. I know I learned a lot. And I think the listeners will really appreciate the stuff that we covered today. So, if you're ever free, I'm glad to do this again. But yeah, thank you so much. Tonight was really great.
Nat, it's my pleasure. And if you get feedback that people say they want to hear more from me, I'd be happy to come back. And please always keep in mind, I do run a public company. So some of the things I said today may be forward-looking statements, and I would refer everyone to our safe harbor clause where they can learn more about our risk factors through our filings with the government at sec.gov. But thank you so much. I've really enjoyed it, and I hope to come back sometime soon.
Very cool. Thank you, everybody, for listening in. All right.